Reading the physiology images, which are frequently asked in the NEET PG and INICET exam. The first image is of Golgi apparatus. It is an electron microscopic image of the cell. On the cell, we can see there is cell membrane, then is SCR, smooth endoplasmic reticulum, and rough endoplasmic reticulum. Towards the rough endoplasmic reticulum side, we have the trans phase of the Golgi apparatus. In this, the plates of the Golgi apparatus. Like it is arranged in the form of plates, so the plates are larger in size, while towards the cis phase, the size of the plates goes on decreasing. So, how we can identify on this cis phase? C is small letter, so on this cis phase, the plates are small, and on the trans phase, tree is a larger letter, so on the trans phase, we have the larger plates of the Golgi apparatus. The next image is about the levels of the T3 and T4 and reverse T3 in cases of starvation. In cases of starvation, as it is visible from the graph, T3 decreases. T3 is decreasing and T4 is almost remaining constant, remaining normal, and reverse T3 is increasing. So in starvation, T3 decreases by 10 to 20% in 24 hours. In the first 24 hours, it is almost constant. Then in the next 3 to 7 days, it decreases by about 50%, with the corresponding increase in the reverse T3. Free and bound T4 levels remain normal. And in case of starvation, as there is no food, so BMI and BUN are decreased. And during more prolonged starvation, RT3 normalizes, but T3 remains low. And in which conditions the reverse T3 is increased and T3 is decreased. These are the condition in fetal life, in fasting, in deiodinases, and in selenium deficiency, burns, trauma, advanced cancer, renal failure, cirrhosis, MI, febrile states. So it will be decreasing in fetal life, fasting, deiodinases, drugs, selenium deficiency, burns, trauma, all the states of stress, burns, trauma, advanced cancer, liver failure, renal failure, MI, febrile states, overfeeding. While on the other side, overfeeding increases the T3 and decreases the reverse T3. So that's all about this graph. Then we have the histology slide of the cardiac muscle. In the cardiac muscle, these are intercalated disc which are visible, marked on by the black arrows in the diagram. These are connecting the two cells. It contains fascia adherens, macula adherens, gap junction, but does not contain tight junction or zonula occludens. It contains both the fascia and macula adherens and gap junctions. Gap junctions are responsible for the simultaneous contraction of the cardiac muscle fibers, and it does not contain the occluding junctions, that is zonula occludens. Then one more diagram of the cardiac muscle. In this, the arrow is marked and there are brownish areas. These are lipofuscin, unstained areas at poles of each nuclei. Unstained areas at poles of each nuclei are visible. These contain sarcoplasmic reticulum, Golgi complex, mitochondria, lipid droplets, and glycogen. Contain sarcoplasmic reticulum, Golgi complex, mitochondria, and lipid droplets and glycogen. And these regions do not contain the lysosome. As lysosome are absent, the lipofuscin pigment is accumulated. Then coming back to the previous slide, so this is the organ of Corti image. Here we can see the three layers, three compartments, that is above is scala vestibuli, then is scala media, then is scala tympani. Scala vestibuli and scala media are separated by the Reisner's membrane or vestibular membrane, and at the junction of scala tympani and scala media is the basilar membrane. Then we have the outer hair cells. Outer hair cells are more in number than the inner hair cells. So inner hair cells are shown here on the right hand side and outer hair cells on the left hand side. Then we have the tectorial membrane. Tectorial membrane is covering the inner hair cells, extending up to the inner hair cells. Spiral limbus, spiral ganglion on the right hand side is coming towards the inner hair cells, and peripheral processes of spiral ganglion are ending at the inner hair cells. Then outer hair cells are supported by the outer phalangeal cells, and inner hair cells are separated by the inner phalangeal cells. Then we have the inner tunnel of Coti, inner tunnel of Coti in the inner hair cell side, and outer tunnel of Coti on the outer hair cell side. So this is about the organ of Coti. Then we have the sleep stages diagram. Sleep stages diagram. Awake stage. In awake stage, we can see the eyes are open. So electrooculogram shows the markings. Then is NREM stage. In NREM stage, we have NREM stage 1. NREM stage 1 has theta activity, and it has less amplitude of the waves. Then is NREM stage 2. NREM stage 2, the EEG is showing the increased amplitude, and NREM stage 3 is more amplitude of the waves. In stage 1 is theta waves, then is sleep spindles and K complexes in the stage 2, and in stage 3 the in stage 3 we have the more increased amplitude delta waves. And stage 4, here also we have the increased amplitude of the waves. Then comes the REM sleep. REM sleep main differentiating point is, in the REM sleep there are waves visible in the EOG. Waves are visible in the EOG. Okay, there's distinct changes in the electroencephalograph, EMG, and electrooculogram. So how we can identify? In the awake stage and in the REM stage, we will be having the EOG pattern, while in NREM stage, EOG pattern will be absent, and in NREM, the waves will be EEG waves will be more marked. And this is the diagram which is commonly asked in the INICET exam. In this, the one most differentiating point is that eyes are open. Here the it has been shown eyes are open, and EOG shows the recording. EOG shows the waves. So this is REM stage sleep. Then we have the retinal layers. Retinal layers from inside to outside we will be studying. First, we have the internal limiting membrane, then we have the nerve fiber layer, then we have the ganglion cell layer, then we have the inner plexiform layer, then we have the inner nuclear layer, then outer plexiform, outer nuclear, layer of rod and cone, retinal pigment epithelium, and on the outermost side we have the choroid. After the choroid we will be having the sclera. So in internal limiting membrane, first starting with the internal limiting membrane, then nerve fibers, then ganglion cells. Nerve fibers will be going to ganglion cells, Then inner plexiform, inner nuclear, outer plexiform, outer nuclear. Then is layer of rods and cones and retinal pigmented epithelium. Retinal pigmented epithelium. Plexiform layers are inside while the nuclear layers are on the outer side. The most important point to be remembered. And ganglion cells are linked to the inside. So ganglion cells will be inside layer. And rods and cones are related to the outside layers. Then next we have the diagram showing the stomach gastric glands. In this there are chief cells, marked and parietal cells, and glandular lumen. Glandular lumen is in between the cells. Chief cells are darkly stained cells, while the parietal cells are pale staining and large cells. Pale staining and large cells are parietal cells. P for pale staining and P for parietal cells, and chief cells are dark staining. Then we have the diagrams of the cerebellum. Cerebellum has three layers: granule cell layer, Purkinje cell layer, and molecular layer. Granule cell layer is compactly packed, then Purkinje cell layer has Purkinje cells, single layer of cells. Then molecular layer has mainly the dendrites and axons of the other cells. So it is fibrous kind of layer. Then one more diagram. The more dark, the more bluish is the granule cell layer, then Purkinje cell layer with a single layer of cells, and then molecular layer, pinkish layer. This is another diagram of the cerebellum. Here also we can differentiate the three layers. Then this is the spirometry diagram. In the spirometry diagram, how we can calculate the FEV1 and FVC. FEV1 will be calculated from the baseline of the waves to the maximum inspiration, FEV1, then in 1 second, and then FVC will be from the downmost point to the above diagram. Okay, total lung capacity is given in the green. Total lung capacity, then residual volume. Total lung capacity minus residual volume is equals to FVC. Then FEV1 by FVC can be calculated, and we can differentiate while it is a obstructive type pattern disease or a restrictive type pattern disease. So that's all the important images of the physiology section. I hope you will like it. Thank you for watching.